Hello, everyone. Thank you to the organizers for inviting me to represent the XChem team in this CCP4 virtual weekend study. So I'm going to share my screen. This time, I hope it works better than this morning. Do you see the full screen? Yeah?
Perfect. Yes, we do. Yeah.
Okay. Okay, so before I start, I would like the pointer. Right, so today I'm going to just talk about the efficient fragment screening on the XChem platform that we have at Diamond Light Source. Before I start, basically, I'd like to just remind the concept of fragment-based drug discovery. There are two ways of screening things in drug discovery. Either you go with a conventional screening where you search for potent molecules. And in this case, you look for complex molecules, which are usually quite big, but the probability then of finding them is pretty low. So you'd have to screen quite a lot of compounds in the order of 10 to the 18th of quite complex molecules. The concept of fragment screening came about 25 years ago, I think, with Aztecs, which basically is a question of deconvoluting all those big complex molecules into small fragments. You're guaranteed to have binding, but they're going to be weak. So the potency will come from a chemical elaboration. So you will have to develop those molecules into lead compounds. The positive things is that instead of screening 10 to the 18th complex molecules, you're going to be screening about, let's say, 1,000 small molecules. So why do we talk about fragment screening by X-ray crystallography now as a primary screen? So why crystallography? Because it's a very sensitive technique. It can detect things in the millimolar range, which is what the fragment's binding affinity would be in the order for. You will probably need structural information in order to progress them into lead compounds, because they are very small and probably not sufficiently evolved to match your site. And why recently are we doing quite a lot of fragment screening by X-ray crystallography is because the throughput has greatly improved. At the synchrotron, you can really now collect data very rapidly. And and the detector technology has evolved so that you really can collect data within minutes. Does it work? Fragment-based in drug discovery. So there are 4 drug compounds in the market, and there are about 40 compounds in clinical trials. So here, so the XChem workflow is summarized on that slide, but basically you will have to put in a project proposal that will be accepted or not, and peer-reviewed panel by the committee. You will have to come with Swiss size 3 drops or 2 drops plates, so SBS format plates. We have libraries that you can use. And then lots of steps have been evolved so that basically you can prepare lots of samples to go onto dedicated beam time on Diamond on IO41. Then the data analysis pipeline put in place is managed by XChem Explorer, and it goes through basically the processing of the data, hit identification through the Panda software, and then after refinement, model dissemination through fragalysis if you want to. So you basically go from crystals in plates, compounds, samples being prepared and collected unattended on IO41, and hopefully the outcome you are getting is a complete mapping of your site of interest. So this is basically a summary of how the program has been taken up by the user community over the years. Since 2015, we've been operational since 2015. And basically last year, we introduced bags, XChem bags, and complete training. And you can see that we've kind of ramped up to 21,000 samples. And actually this year, due to COVID, we're a bit lower, but not so much. So the whole pipeline seems to work and is pretty resilient even despite COVID-19. Right, the strategy that we've put in place, having been through so many projects, we've realized that because beam time is precious, if we need to fail early, or at least we need to evaluate the crystal system early. So we've divided the experiment into 3 steps. The first one is solvent test. Basically, you evaluate your crystal system for tolerance to DMSO or ethylene glycol, because we have the libraries in those solvents. At the end of that step, this step basically uses about 30 to 50 drops of crystals, and we test 4 conditions, DMSO conditions or ethylene glycol conditions, and we do 2-time course, 1 hour or 3 hours. Basically, at the end of that test, what we want to know is the integrity of your crystal and whether they still diffract in the presence of that solvent. So if it works, we go into the pre-screen. If it doesn't, we fail it and we look for a different crystal system or a different way of dealing with it. Basically, you need to think harder. In the pre-screen step, we're gonna take forward the condition that we found in the solvent test, and we're gonna put it with compounds. This is basically because we know that sometimes compounds affect how the system is behaving. So that again, at the end of that experiment, which takes about 100 to 150 drops of crystals, you should know whether your crystal system really tolerate the compounds in that condition. And in principle, you should already start to see some initial hits. So it's worth stopping at that or pausing at that step to really look through the whole thing and check that with the about 200 experiments, you know already that either it's gonna work or it's not gonna work. And if it doesn't work, it's better to stop here and look for a different crystal system and then go back to the start. While if it works though, then we're pretty sure that we can basically do the rest of the screen pretty rapidly. And hopefully the outcome, as I said, is a mapping of all possible binding pockets that you have on your protein in this crystal system. Oops, sorry. So we have a lot of fragment libraries in-house now. There's the DSI Poise library that has a fast follow-up chemistry that was basically derived in 2017 by Oakley and between Diamond and SGC. We currently also have tested recently with the COVID-19 the EU Open Screen Library. So this is basically a European consortium and they provide 900 plus compounds that you can use. And they also can provide you chemistry consulting possibility. This is quite crucial. So the idea behind all the libraries that we are providing is that you can do the follow-up quickly. We know we can screen. The most important is whether you can follow up afterwards. So the DSI Poise library was designed for that, and the Open Screen is providing you consistency so that you can continue in the chemistry if you don't have that in place. Then the other libraries we were very interested in, we've grouped them into probing libraries. There's the FragLights. Basically, these are very small compounds, less than 300 Dalton. And the idea is that we would look for hotspots of pharmacophore. The FragLights, basically, they're going to be even weaker binders. And in each of the libraries, there's a way of seeing them. The FragLights has some highlights that you can use to identify them. And the mini frag, basically, you have to soak in extremely high concentration. And I think we have the soak at 1 molar now, concentration. And then there's the York 3D library that we've been assessing also as part of the probing library, which are more 3D-like. And then we have the Covalent library from near London that we've used quite a lot now on the viral proteases in COVID-19. Of course, you can also use your own library as long as they are in the ECOPAN compatible plates. So what are you getting for the sample preparation? There's a lab, oops, here, with 2 ecosystems. which are basically acoustic wave dispensing liquid handler, and 3 shifters here, 1, 2, and here, 3, which is basically an XY stage where with a sophisticated software to record what you're doing during your harvesting steps. So the process is basically, you come with your plate, or we grow your crystals currently. We image your plate. This step is to select which drop you want to use for the experiments and where you want to dispense the either solvent or compound. And we're recommending, of course, not to dispense directly on the crystals, because the echo is quite powerful and can destroy your crystals, but next to it, so that there's a slow diffusion into the drop. We then dispense the compounds using the ecosystem here. The harvesting with a shifter, and everything is registered into I-SPY-B for unattended data collection on IO41. Everything is, of course, talking to a database so that you can keep trace of all what you're doing. So in terms of data analysis, the pipeline is basically controlled through XChem Explorer, the software that Tobias wrote. And basically it consists of several steps. The first round is to build your ground state model so that you have a reference model of without any ligands. And we then use Panda to look for weak binders. So the Panda software is equivalent to like a background correction. It uses the average of most of the maps that you have where officially there's nothing in, and you subtract that to each individual electron density that you have in order to see weak binders. And then when you've done the hit identification, you've placed them, you've refined them, you can also deposit everything using XChem Explorer. So the typical fragment screening campaign output that you're getting. So I've just taken the example from the main protease because we've worked on it quite extensively recently. So you'll have basically everything in the structure. Sometimes it'll be intrinsic sites like this one where it was the active site. You'll have probably some novel sites. For example, those ones are in the binding, in the dimer interface, and could be some allosteric sites. And you'll have the green ones where basically they are all over the place, usually in the crystal packing. So the crystallographer has to review those data, has to assess whether those sites are useful or not, and prioritize, of course. We tend to have a hit rate that varies between 1 and 30%, depending on the project. And for this is basically an example of what we've done during the COVID lockdown period between February and September. So we've tried to do quite a lot of fragment screening campaign on antiviral targets of the COVID-19. So our workhorse is basically the main protease, where now we have compounds in the low nanomolar range that hopefully are going to be useful. At least we can take it to the clinical phase. Then a lot of those targets have been then partnered with an international collaborator, for example, the NSP3 microdomain. So our health group in Oxford is now working with this UCSF group, which provide basically their expertise in ComChem as well. The NSP13, as far as I know, has been partnered with some industrial so that they can follow up the different hits that they are interested in. And as you can see, we know we can screen and we know that we can find fragments. So with that, I think I'm within my 15 minutes. And I'd like to thank the whole group that have been putting in place this platform. It's a collaboration, extensive collaboration between Diamond and the SGC groups from Frank. on the SGC side, at the Diamond site. And if you have any questions on access or requirements for the academic, you can talk to us, while the industrial will contact the industrial XChem team. There's a lot of information on the web. And also you can access that platform through the Agnex Discovery, which is, we've used it before the lockdown, and it's very helpful for European users. And I thank you for your attention.